In our body, there are thousands of reactions happening all the time in our cells. These reactions are controlled by enzymes, proteins that act as biological catalysts. Here's a definition that you need to know about for your exam. Catalysts are substances that increase the rate of a reaction without being used up or changed in the reaction. So enzymes are biological catalysts. They speed up the rate of reaction in our bodies and reduce the need for them to take place at a higher temperature. After all, our internal body temperature is only 37 degrees Celsius. Enzymes catalyze the reactions in our bodies, working both inside and outside the cells. Here's some examples of some enzyme-controlled reactions. DNA replication, where enzymes help copy a cell's DNA before it divides by mitosis or meiosis. In digestion, enzymes are secreted into the digestive tract to help break down different types of food molecules. Protein synthesis, enzymes here help form the bonds between amino acids, making a polypeptide chain. Enzymes are proteins. Each different type of enzyme is made from a different sequence of amino acids. These amino acids fold to determine the enzyme shape. A key feature of an enzyme structure is its active site. If the active site changes shape, the enzyme will no longer work. Let's take a look at why this is. Only a particular key can open a particular lock. If the key isn't the right shape, then the lock won't open. This is a lot like how an enzyme works. Each enzyme only works on one substrate. The substrate is the molecule or molecules the enzyme is acting on. For the enzyme to work, the substrate has to fit into the active site. If the substrate doesn't have a complementary shape to the active site, then the reaction won't be catalyzed. Because of this specific shape that's needed, this is described as the lock and key mechanism. Changing the temperature can affect the rate of an enzyme catalyzed reaction. As the temperature increases, so does the rate of reaction. This is because the molecules have more energy, enabling them to move around faster, which increases the chance of them colliding with each other and reacting. If it gets too hot, the bonds that hold the enzyme together can break, causing the active site of the enzyme to lose its shape. The substrate can no longer bind, so the enzyme is described as being denatured. We can show this happening using a graph. As the temperature rises, the rate of an enzyme catalyzed reaction increases. However, at a certain temperature, the reaction starts to get slower, even though the temperature is still rising. This is because the enzymes are becoming denatured. Every enzyme has a temperature that it works best at, called its optimum temperature. Can you estimate the optimum temperature for this enzyme? For this enzyme, like most enzymes in our body, it's 37 degrees Celsius, our body temperature. Enzymes also have an optimum pH. The pH, whether it's too acidic or too alkaline, can also affect the bonds in the enzyme and can cause it to become denatured. Take a look at this graph. Which enzyme works best in neutral conditions and which might be found in your stomach? A lot of enzymes work best at pH 7, which is neutral, like enzyme B in the graph. Some can work in very acidic conditions, such as those involved in the digestion of protein in our stomachs. They can work at a pH of 2, which is extremely acidic, like enzyme A. So what does this graph show? An enzyme has to collide with a substrate molecule in order to catalyze a reaction. This is why the concentration of substrate affects the reaction too. If we keep adding substrate molecules, then the rate of reaction will increase, as there are more substrate molecules that enzymes can collide with. At this point, even if we add more substrate, the reaction can't go any quicker, because all the active sites of the enzymes will be occupied.